The overall goal of this procedure is to demonstrate the use of a lateral flow device for the rapid detection of Aspergillus diagnostic antigen in human serum and BAL fluids. This is accomplished by first applying the serum or BAL sample to the release port of the lateral flow device. The second step is to allow the solution to migrate along the membrane. The next step is to determine whether the test is valid by examining the internal control line. The final step is to interpret the test result by examining the test line. Ultimately, the lateral flow device is used to detect the presence of Aspergillus diagnostic antigen in serum or BAL fluids.
The Aspergillus LFD can be used as an adjunct test for the diagnosis of invasive pulmonary aspergillosis. The main advantage of this technique over existing methods, like the Aspergillus galactomannan enzyme immunoassay, pan-fungal beta-glucan assay and Aspergillus PCR test, is that it is a rapid point-of-care diagnostic test requiring minimal laboratory facilities and training. Though the LFD has been developed for the detection of Aspergillus species, lateral flow technology can also be applied to the detection of other infectious pathogens, such as Scedosporium, Fusarium and Rhizopus species, by incorporating monoclonal antibodies specific to these organisms. Visual demonstration of this method is critical, since interpretation of LFD results requires familiarisation with the assay format.
To collect serum from untreated blood samples, allow blood to clot at 4 degrees Celsius. Serum and brochoalveolar lavage, or BAL fluids, should be stored as aliquots at -20 degrees Celsius prior to use. Storage as aliquots limits the potential for degradation of the target antigen.